Thank you very much for the introduction. And thank you very much for inviting me to speak to this meeting. It's a great honor to speak in this track. And I hope I have time to give you an overview of the work we've done. I mean, it's really about the evolution of CATH, this talk, and also the communities that we interact with, the tool developers that we sort of share our knowledge with, that we work with, and who also use our data, but also some of the experimental communities that we've worked with as well. And if I have some time, I'll present a bit on how we use the data in CATH. I should say that, well, actually, congratulations to BOSC as well for so many years. And this year we're celebrating 30 years of CATH. We have a bit of an existential crisis because we have funding for 2 people in the team, and Wellcome Trust, to fund one of those people, have decided not to fund resources anymore. So it's even more necessary for us to collaborate and seek funding elsewhere. And I'll tell you a bit about how we've managed to do that. Just for those of you less familiar with, and I'm sure many are not using structural data in the audience, we've always used structural data. We've always worked at the domain level because we feel this is a fundamental unit of functional evolution. And I guess our community are experimental biologists and structural biologists. They want to know what kind of structure does their protein have. But more importantly, they want to know how does it use that structure to perform its function. And you can see that there's a nice picture of the polypeptide chain here, but it's not till you put the atoms on that chain that you can really sort of see the pockets in the protein. You can see the chemical characteristics of the residues that interact with the compounds in that pocket. And you can start to understand how the protein does its function. And this is something I did enzyme kinetics as a PhD student. I hated doing the experiments, but I've never lost my kind of passion for understanding how proteins function and also how they evolve different functions. And so I was very lucky that I came in at a kind of fairly early stage. And this was a point where a great pioneer in this field, who you heard yesterday, Janet Thornton, realized that the number of structures were really increasing thanks to crystallography techniques. And really, the evolution of CATH is about trying to keep up with the data all the time, the new data that comes along. So Janet foresaw that this crystallographic data was really going to expand. And at the time, I was working with a brilliant scientist, Willie Taylor, at Mill Hill. And he was interested in developing algorithms to superimpose protein structures to see how related they were. So I worked on this algorithm with him, and this is obviously a perfect tool to look for evolutionary relationships between. Sorry, I'm sure you don't need to hear me twice. I'm trying not to stand so near. Oh, I don't know if that can. I'll try and speak slowly. Anyway, so I went to work with Janet. Sorry. No? Okay, great. And that was the beginning of CATH, really. And we worked on algorithms to decompose structures into their constituent domains and then compare the structures and classify them into evolutionary superfamilies. And this is just really a representation of the value of structure. So I'm showing you in this picture relatives that have very low sequence identity. So you couldn't spot these relationships using sequence information, but the structure is a fantastic fossil of the evolutionary record. And you can see the blue structural core, which all these distant relatives have. But you can see that there are copies of human relatives here. And quite often the duplicated copies within an organism, the paralogs, diverge structurally to have somewhat modified functions. And this is what fascinated me. How do these structures diverge? How do they evolve new functions? And this is obviously very important for our communities. They want to understand, does the new structure they've solved have the same function as other relatives in the superfamily? And at this point, I want to acknowledge Ian Silito, without whom I'm certain CATH would not exist. He's our key research technology professional. He's been promoted by UCL to, I think, what's equivalent of associate professor at UCL, and he's really our director of FAIR. He's the reason everybody can look at CATH data, use CATH data, and he's here if people have questions afterwards. Thank you for applauding. He's the reason. He's kept it all going. And actually, just to highlight that he's he's done the websites, he's done the database, he does everything that make it fair. Every iteration of the web pages. Actually, this is the kind of current one. And the reason we don't have more is just we need more funding, because Ian's had to do more and more work to cope with all the extra data. And we obviously provide overviews of the tools, and we have a GitHub. And again, please contact Ian if you have any requests of modifications to our resources. So I want to talk now how we had this structural data. It was very limited, really not very much structural data at all, because crystallography techniques are slow and it's difficult to get structures very, very frequently. Actually, I should have said this sooner. We've always felt it was important to collaborate with our competitors. I think someone else mentioned that. And we've been very fortunate to work with, I think, and Nina is here, Tony Andreeva from SCOP and Alexei Mertzin, who've been fantastic collaborators. Actually, we started with the eFamily project funded by the MRC. We've had a number of BBSRC projects since then. And more recently, we got funding in the 3D Scaffold project to actually archive and update SCOP continuously, and that's happening at the EBI. So we're very proud to have been able to work with, and Cyrus Chothia, who sadly passed away, who's really a pioneer in structural bioinformatics. So I want to reach out and thank those. This was us celebrating 20 years of CAS and SCOP, and then this year we will join and celebrate 30 years. But I want to move on to the new data that we had to cope with. And this was even more massive wave, fantastic expansion of sequence data, thanks to the sequencing initiatives. And we really wanted to tackle this, because many biologists would come to us and say, well, you don't have the structure I'm interested in. My protein doesn't have any structure in your resource. Do you have any idea what family it could belong to? And we were lucky in that, I mean, again, we've been very lucky with the tools that have come out from the community. So Sean Eddy developed this fantastic hammer tool, which Interpro is very widely used. I think that's under threat of funding at the moment as well, but it's an absolutely key resource for mapping structural data onto sequences, for predicting which structural family does a sequence belong to. So we could collect information from our structural relatives, build hidden Markov models from those, and use those hidden Markov models to then pull in all the UniProt sequences into CATH, as many as we could. And about 50% of the proteins in UniProt could come into CATH, which is sort of reassuring, because it meant that many people thought that CATH and SCOP were biased. You know, that biologists had focused on just some key areas, and these families didn't relate that widely in nature. But we could see by bringing all the sequences in from UniProt that actually really large part of UniProt was covered by structural families. But the more important thing was that by bringing in, and this expanded CATH 200-fold with new relatives in each superfamily, this brought in huge amounts of functional information as well, so we could start to understand what were the functions of the families. And again, this necessitated new websites. I want to thank John Lees, who's done an awful lot of work on Gene3D and made this data available for the community. I also want to say that having this information meant that we had new collaborators, new groups that we work with, and we were invited to become part of. I think we've been in Interpro for maybe nearly 20 years now. And this has helped. I mean, I guess I'm convincing all of you the importance of collaborating with your competitors, and by bringing together all our information, we help the biology community. They can see the different predictions that we all make. They can start to understand which parts of the sequence are difficult to annotate, which parts do we all agree on. And also, it's just great fun to work with the other people working in a similar area. You can share your ideas on ontologies, you can share ideas on how you're solving challenges, and you all bring different perspectives. So you really learn a lot from each other. And I have to say, the Interpro projects that we took part in, and we've been involved in several, have been immense fun for kind of discussing how we can take our algorithms forward and how we can share our data. And I should say that we work closely with a lot of Elixir initiatives, and Elixir-supported platforms like BioTools and bioschemas. And again, Ian's the person to ask if you want more information of what we've done in that area. We were also lucky to start, as we brought in these sequences and we annotated them with structural information, we realized that, a lot of our data and also a lot of our interests coincided with people predicting structures from sequence, so homology modeling, ab initio prediction. And we then got together with that community, and we were, again, lucky to get funding. Scott and Tony, who's here in the audience, were involved in that work as well. So we had a new collaboration where, again, we could start talking about what are the structural ontologies that we need to develop to make sure that we're all speaking a common language. And these were the groups that were involved in that. And hopefully you can see on this page the value of bringing together the data from these different teams. You can see for this particular protein, there's quite a lot of agreement in some parts of the sequence, but you can see there are some regions where we have an extra blue domain that other people haven't detected. I think consensus work when you're using algorithms is essential. None of the algorithms are perfect, and you need to be able to compare the predictions, compare the data, and work together on the challenges and on the ontologies. So it's been a great privilege to this is just and I should say again, this was Ian that developed all these lovely web pages, and the importance of presenting your data in a very visually intuitive way. Again, we've worked with these teams to develop research data management kits to provide advice to the community, especially for homology modeling. And again, it's very important to get your agreement across the community on your ontologies. So bringing all this data in from the sequence resources made us realize that there's really a lot of bias in the evolutionary families. I mean, some evolutionary families are absolutely massive. So 100 out of 5,000 superfamilies account for more than half of the data, actually. So these are superfamilies that you find them in all organisms of life. Some of them are massively duplicated within some organisms. And it's these families that I'm most excited about, to be honest, because it's these families that diverse enormously in their structures and their functions. And we can learn a lot from these families about how to design new proteins, how to understand whether some genetic change altering the structure might affect the function of the protein and perhaps cause a disease or enhance resistance to a drug. So analyzing these families has been really probably what excites me more than anything. And we were very fortunate, again, to be involved in another collaboration. And this was a massive project in the States. So at the end of the Cold War, they had a lot of money. They didn't know what to do with it. And so they decided to put it into environmental research, but also into genomes research. And we were very lucky to be part of the Chicago Center in that. What they wanted to do was to take all the sequence data in UniProt. They could see that the structural data mapped to probably less than 5% of all that sequence data. And they said there were 4 bioinformatics groups. Burkhard Ross was another one, and Adam Godsick. Which ones should we target? We hope to get about 5,000 structures at the end of this. Which ones should we target? And we started targeting things that we thought were completely novel. And we found that quite often they weren't. They did belong to families we'd already seen. So then we decided, actually, what we really need to do is to dig into these mega families and to get structures for things that we predict will be functionally different. And so how did we do that? This necessitated a completely new algorithm where we just worked with sequence data. And we took all the superfamilies, actually, and we first of all clustered by sequence information, very highly similar things. We can then build patterns from those highly similar things and use the lovely, wonderful tool of Johannes Soding, HH-Align, which allows us to then merge clusters that are highly similar in their sequence patterns. And a major part of this algorithm is deciding when not to merge clusters. And because we're interested in functions and functional sites, we don't want to merge clusters that have different functional sites. So different patterns of conserved residues that are likely to be important for function. And you can see from this picture where I'm trying to decide whether these two fams should be merged, some positions are completely conserved because they're important for making that structural core I told you all evolutionary superfamilies have. But other positions are conserved, but in completely different ways. Here you have a positive arginine, that switch to a negative serine. So these functional families are doing something really quite different. And it's these differentially conserved residues that allow us to find these functional families. Just an example on this slide. This actually is a family where it doesn't structurally diverge a lot, but it diverges a lot in chemistry. And you can see two different FUMFAMs. You can see actually the active sites are typically in the same position. But if you look at the right, at highly conserved residues in structurally equivalent positions, you can see sometimes there are similar residues that are involved in packing of these proteins. But you can also see in color the catalytic residues at equivalent positions. You can see significant changes in the chemistry from a negative aspartate to a positive histidine. So these tools, and I should also highlight Mona Singh's work, because we use her wonderful group sim algorithm to help us with these types of analyses. And I should at this point also mention that we've taken these tools forward with these new developments in protein language models. So we collaborate with the team of Burkhardt-Rost, and I want to reach out and thank that group as well, because they're experts in protein language models, generating embeddings, and we've been able to use their wonderful algorithms they transform the protein sequence into a vector of characteristics that actually for each residue, you get information on the structural context of that residue. So these are more powerful than hidden Markov models. And it turns out they are faster as well. I want to also highlight, I will show another picture later on, Nicola Bourdin. Again, he's here if anyone's got any questions, who has really been driving the development of these FUNFAM algorithms, bringing in the embedding data, working in many ways, which hopefully I'll have time to highlight later on, to take this algorithm forward and means we can cope with the data. So we've switched our method to using these deep learning language models and embeddings. And I should say that they're not perfect, these methods. I mean, this is a really hard challenge. And again, I want to reach out and thank the CAFA community, because I think without that community, there would be no way of evaluating how well we're doing at function transfer, function inheritance, function annotation. And you can see that actually, so our recent, which is much faster embedding-based method, is comparable to HMM. It's slightly less sensitive, but it's much faster. But you can see that it's still not perfect, because what I'm showing you here is the number of functional families that have a single functional annotation, which is what we want. And you can see that some families have got multiple annotations. The problem's not solved. This is a very difficult challenge, segregating functional groups. And it's something that's a key kind of research area in my group. But you can also see that when we get it right, we can see that these structural relatives within a functional family have and sequence relatives. So they're even with very low levels of sequence identity, where we do have structural information to confirm it, we can see that they have very, very highly similar structures. So form follows function. And that means that we can capture information about the binding pocket. And if we have a protein for which we have no structural information, we can still transfer information about the likely structure of the binding pocket, and even down to the conservation of the ligand binding poses as well. So this is a really key area of research in the group. How do we identify these functional families? What are the characteristics of the binding pockets? What is their likely biochemistry in the cell? And again, just to reach out, I think Ido's in the room, and to thank CAFA for the great work. Actually, we didn't take part in CAFA 5. We weren't quite ready for that. We were very busy with alpha folders, you'll see in a minute. But we've done reasonably well at CAFA. You need a low score for CAFA. And this is a slide that came from the CAFA 3 competition. But as I've shown you, there's still really a long way to go with function prediction. It's a major challenge for the field. We've tried to excite John Jumper and DeepMind in this, but I think they decided that it was too difficult, and it's too hard to assess when you've won. And that's where CAFA does a great job, because it assesses according to lots of different metrics. And just to make another important point, that by grouping things together, having very high similarity in differentially conserved residues, we get very good multiple alignments, very good conservation of structure, and these key highly conserved residues shine a light on the functional pocket. And we can start to think about, because structure is wonderful, but we discovered in the structural genomics initiatives, one of the big criticisms was, you've got the structure, so what? You've no idea what it does. But actually, if you combine the structure with evolutionary data, you start to see where are the key pockets, what are the key residues, what could it be binding. So this is why it's a major focus. And again, thanks to Ian, we needed a whole set of new web pages. We needed a whole set of new workflows. Each time we bring in new data, we have to really rework everything we do. So now I want to talk about another collaboration. And this time it was a group actually on our doorstep. And this was to handle this massive new wave of data. Huge amount of high quality structure predictions from AlphaFold. You heard from John Jumper last night how that evolved over the years. And this has been a really exciting and actually, Nico Bourdin, who's done a lot of work on this project, but all the data from AlphaFold came out on his birthday. So, and he lost his summer holiday, and but it's also been the most exciting thing to happen in his career, I think. So we were also very lucky as a community that this amazing tool by Martin Steiniger in South Korea. And we worked with him and Stephanie Kim to find out what thresholds we should use for identifying structurally proteins that were similar enough to be homologs, or proteins that just had some core fold similarity, but we didn't know whether they were homologs enough. So it was a real privilege to work with them. And their algorithm is using deep learning again. It's transforming the structure into a linear string, and you could use amazingly fast algorithms. So it's just game-changing, 10,000 times faster than any other method that was available at the time. And I think they worked closely. I think they're on the AlphaFold paper, because without this tool, we would not be benefiting from all that AlphaFold structural data. So really thanks to that team. The other problem you have with the AlphaFold structures is you can see there's lots of kind of disordered spaghetti-like bits. And what you really need to do is find, for us, we needed to find the domain regions. And none of our tools were fast enough again. We need to completely rework all our tools. We were lucky because we developed this method with Chainsaw. We had a very talented PhD student in the group, Jude Wells, who worked with Nico and others in the team to develop this algorithm. And it exploits the structure, the contact map of the AlphaFold structures. So you can see from these contact maps which residues are in contact with each other in the protein structure. And you get very distinct patterns. So it's a bit of this is slightly simplified, but you get a lot of contacts within the domain, very few contacts between domains. You get a very clear pattern, and you can use convolution neural networks. And we were lucky we used tools adapted from David Baker's wonderful suite of tools. What's nice about these algorithms is you can distinguish what we call continuous multi-domain proteins, and more than half of proteins in nature are multi-domain, from what we call discontinuous. And this is what happens when, during evolution, you get a lot of kind of genome or DNA shuffling, if you like, during evolution. And that's how you get lots of different domain combinations during evolution. And sometimes you get shuffling of genes within another gene. So in this case, you can see this red domain has been inserted within the blue domain. It's really difficult to find these instances by sequence-based techniques, but structure shines quite a good light on these, and you can see actually this contact map pattern is very different than from a continuous multi-domain protein. So these techniques were very helpful in segregating these different types and also identifying the domain regions. And again, we did use a number of other features. I won't go into detail because it's all published. And as with all our tools, we try to benchmark them against, and this is really essential, against other cutting edge algorithms at the time. So we benchmarked against Unidoc, which was one of the key algorithms at that point. And you can see that for multi-domain proteins, that it performs really well. I also want at this point to say something about the PUU algorithm, which we used for years by really brilliant bioinformatician Lisa Holm. She also developed DALI. Many of you may be familiar with that. So again, she's was a real pioneer in this field. And her algorithm is wonderful, but it's too slow to use. So with these tools, we had a very fast and accurate method for chopping domains. We had Martin Steiniger's wonderful method for comparing domains. And we set about tackling this massive database. How will it change your understanding of how structures and functions evolve? And this is where we embarked on another collaboration. And this was actually a team that we're in our open lab at UCL, in the Darwin lab at UCL. So they were right on our doorstep. And David Jones is a a pioneer in structural prediction, actually. He worked with AlphaFold on the first iteration of that algorithm, but since the development of AlphaFold, he's become interested in new challenges, fast ways, a bit like Martin Steininger, fast ways of scanning AlphaFold. And for that, he needed a chopping algorithm, he needed a search algorithm. So he developed resou to chop domains, and he developed resou search to search through AlphaFold. And we compared chainsaw and resou. They're very complementary. Resou tends to overcut, we tend to undercut. So we thought, well, it makes sense for us to work together. And it's been, over the last couple of years, very intense experience, but very, very wonderful learning experience working with that team. So on the right, you can see, actually, again, here's a better picture of Nico Boydin. And also want to thank Jude for Charlie and Ian as well for their contributions to that project, and Andy and Sean from David's team. And again, what we did, which is important, because I mean, these algorithms are just not perfect. This is just the beginning for these very fast chopping algorithms. And we're working on a new iteration of this algorithm at the moment. We can see all the things that aren't quite right about it. So we used 3 algorithms. And where we had at least 2 algorithms agreeing, then we felt reasonably confident that the choppings are probably correct. But we know that there's still work to be done on this. And we could see then that we could take these non-redundant structures, we could get nearly 370 million domain structures. It's still a massive amount of data for our teams. We barely had the resources to cope with it. You can bring together highly similar proteins with more than 50% sequence identity. They certainly have the same structure. And then once you've done that, you can look at these clusters and see whether they're similar to anything we have in Cath. And we did that using Martin Steininger's amazing algorithm. And we could see that nearly 80% mapped to Cath. Now, that's interesting because these are all UniProt. And I told you that UniProt using hidden Markov models, 50% mapped to Cath. Now, with this structural information, 80% matches to Cath. And again, that emphasizes the importance of structural information to detect these very remote homology relationships. And this was a massive expansion of Cath, and it's involved many. Well, I'll get into that in a minute. Some of the AlphaFold structures aren't perfect, quite often where they don't have deep enough alignments or where there's lots of disorder. So anything that didn't map to Cath, a lot of it actually was highly disordered. And even the secondary structures weren't packing properly, so we could remove them. And after that, and I should say that actually David Jones was really, he wanted to find out what are the really novel folds. So we used his resou search algorithm, we used FoldSeek. We really intensively searched anything that was remaining. Could we bring it into Cath? We brought more stuff into Cath like that. What's left over? How many novel folds are there? We found these beautiful, highly symmetric structures. The reason these don't map into Cath is because although they're similar to what we've seen before, so we have a 6-bladed and 7-bladed propeller in Cath, these are repeats. We don't have 11-bladed propellers in Cath. So we have something like it, some architectural arrangement like it, but nothing with this number of repeats. So these beautiful symmetric repetitive structures didn't come in because there were just more repeats than we'd seen before, but they are in some senses novel structural superfamilies and arrangements. And we found lots of highly irregular things, 7,500 new folds. But also what's exciting for me, many things come in because they have a similar folding arrangement to what's in Cath. But we can't detect any evolutionary relationship. So these are totally novel superfamilies, and we will need another few years to really make some sense of those. So the other, I think, useful information, again, for the biology community and for the prediction community in particular, is that we found lots of new domain combinations in UniProt that we hadn't seen before. Domains that are fused together that we haven't seen in the PDB, that we weren't aware of, and sometimes similar domain combinations, but completely different ways of packing the domains together. Now, this data will be useful for the community that are predicting protein complexes, because it's telling us something about the ways in which proteins pack together, and it will help guide those communities in their algorithms. We also saw that the families, the mega families, were massively expanded by this data, and some of them like 10,000-fold. And what was particularly exciting were alpha a lot of alpha membrane associated proteins that are hard to solve by crystallography, and some of these drug efflux transporters. So these pump drugs out of bacteria, and there were very few structures, and we could find many, many more of them in TED. So this is just a kind of summary 400-fold expansion of Cath, completely novel folds. And I think for me, what's exciting are these new ways of doing functions, these new structural superfamilies, which we haven't, we're just starting to dig into. And again, thanks again to Ian, more beautiful web pages from him, but also thanks to Samir Valenka and the team at the EBI, because the AlphaFold database is now disseminating the data through their web pages as well. We've made all the TED data available for anybody who's interested in using that information. Please, I know actually, thanks to Tony and who's now working with PFAM, they're using the data and they're very kindly feeding back all the things we get wrong so that we can get a new version of the algorithm. And big thanks to Samir for helping us use the AlphaFold data. And at this point, I just want to mention that thanks also to the Elixir initiative that is promoting kind of fair processes and integration of data from different groups and promoting collaborations between different groups. So we'd worked together with a lot of structure prediction providers I showed you through Genome 3D initiative. And now with AlphaFold and actually RosettaFold that David Baker's group came out with shortly afterwards, there's a lot of high quality structural information for which we're providing domain annotations. And Samir Valenka at the PDB, first of all, with some Elixir funding that we managed to get, we started working with the Swiss model group, which also have a wonderful resource providing structural predictions. They're refining AlphaFold data and providing wonderful annotations for that data. So this 3D beacons that Samir has set up will grab predicted data from all these different resources, from AlphaFold, from Swiss model, from RosettaFold, from any other groups that generate models that aren't in AlphaFold database, refined models. These are APIs that will bring the data together and integrate it so the community can see differences between the predictions and can use as much data as possible. And again, just to show you some pages. And also just to mention that these are very old, Ian will recognize these, from like more than 20 years ago, but TED has meant, and actually I've shown this picture, it's one that Ian likes to show us to slow us down and to make sure that every time we have new data, every time we have new algorithms, we really have to try and capture that properly. And actually, it's been, to be honest, it has been exhausting the last year. We've had to completely rework all the algorithms that Cath has used and the way we organize the data. It's such a massive expansion in structural data. And we're currently working on Nextflow workflows, trying to make sure that all our algorithms are modular, that we can also bring in new algorithms that the community develop, and we can make sure everybody can use our algorithms to chop their protein structures and classify their protein structures that might not be in our resource yet. So I don't know, have I got just a couple of minutes? I could just very quickly talk about how well, sort of, first of all, I just want to explain how I think structured data combined with evolutionary data is key to understanding function. And this is just an example of, these are proteins that are very important in bacteria. They're acetyl transferases. They're often targeted by drugs, but many bacteria have no drugs and nobody knows what family their proteins belong to. And what we could see through using the TED data, first of all, using our functional families, we could see when you look at the experimental data, there are two clear functional families in this superfamily of these proteins. You can see in tan, and you can see, hopefully just about see the structural core there, but you can see one fam has this structural decoration here at the top. The other one has structural changes here on the left. I'm a bit dyslexic, but you can see on the side here, structural changes. And there's a massive expansion of this structural data thanks to AlphaFold, 1,000-fold expansion. And what's important is that, we're just for an example, we took some pathogens for which there's very little information at all, showing on this slide. So these hydrotate worms that cause huge problems in Africa and no drugs designed for at the moment, and botulus, also a serious problem. And we could see that by taking the structures from AlphaFold and from TED and scanning them against the experimental structures, that they belong to this family, and we could see which functional family they belong to. And that's important because the different functional families are involved in different oligomerization states. So the tapeworm, it's actually a monomer, but during evolution, there's been a fusion with a different domain here. And you can see that the ligands bind in between the two domains. Whereas for the other functional family, you have a trimer of single domain protomers. And again, you've got a completely different functional site. It's between the monomers. So different functional mechanism for these two different functional families. But also what's important is when you bring in the highly conserved residues from the functional families, now you know much more about the binding site for the compounds that these proteins bind. And what I'm showing you in pink is the residues are conserved across the whole functional family, and there are very many relatives in this, but it wasn't known before that this tapeworm belonged to this family. And here I'm showing information on a compound that's been designed. You can see that the tapeworm does have some differences shown in blue, but this information on the active site and the key residues will help in either repurposing the drug or designing new drugs for this tapeworm. And on the right here, you can see the same thing for this botulus pathogen. Again, the active site, you can see the FunFam highly conserved residues in pink. And you see already you have very key information for how to either repurpose the drug that is there from the experimentally characterized relative or to design new drugs. And just if I have time, 1 minute to highlight, I do want to highlight this community because I just want to thank Rob Fenn for this amazing Magnify resource, because UniProt has got, hundreds of millions, but Magnify has got billions of sequences. I mean, it was important actually for some, these types of resources were important for the deep learning that the protein language models do. And we've been working with Rob to trawl through this data and to find new relatives of a plastic degrading enzyme. So these plastic degrading enzymes belong to this alpha beta hydrolase superfamily. And what we did was to work with Rob. First, actually, at that time, we started using hidden Markov models. So we took the structurals from the superfamily in Cath. So these proteins have evolved from chitinases that you often find in insects to degrade, the polymeric material in leaves, fibrous material. And obviously, they can also degrade plastic polymers. What we did was, and here I want to reach out and thank Nico Boydin in the group, because we had to totally, again, rework our tools because it's massive data to search through. So we parallelized our FunFam algorithm and I think I've mentioned before, we also used the PROS to make it faster as well. So Nico Bourdin was responsible for bringing the information from Magnify, into Cath. And you can see that we were able to find some really interesting functional families. And this way have lots of relatives in them, which means that these types of structures have remained through multiple generations. They're from the sea. All of this is marine data. And by using our strategy, we could then sort of compare across these different functional families that you find in the sea. So each functional family represents a new way of evolving to bind plastic, a new way of doing chemistry to degrade plastic. And so we wanted to compare across all these different ways of degrading plastic. Could we find what are the key residues that are changing? Because each of them will have some different residues in key positions. And so we could use our FunFam strategy. We found actually 22 residues that in the binding pocket that look really key. They're changing in different ways. They all map to the binding region. And we use this data, these sites, to then make a tree of relationships from all this marine data that we had. And we did this because actually, and this is a problem, our experimental group could only test 11 of them. And we had like thousands of sequences that we wanted to try and analyze, but they could only test 11. So we used information about differences in the key residues to choose 11 possible ones to analyze. And fortunately, 6 of them do degrade plastic. One of them looks really interesting. And what our experimental collaborators will do is subject them to even more rounds of evolution artificially in the lab to see if they can work up a more act, because it's a very sluggish enzyme. It's a pretty useless enzyme in the sea, actually, because obviously bacteria don't totally depend on plastic. So we have to accelerate the evolution of that enzyme. So I'm sorry if I hope I haven't gone over too much, but I want to point out that it's very, you heard also from a fantastic talk from Amos this morning, it's really, really challenging to get funding for data resources. As I said, we've only ever had 2 posts throughout. And the reason we survive is because we collaborate with other tool developers who we really love working with, actually. We can share our challenges, we can share our solutions, but also we work with experimentalists. And that community are really important to us too, because they're the ones that tell us, this is what we need CAS for, this is what, it's the reason we started developing our farms, because people wanted to know what are the changes in structure that changes in function. And now we have the privilege of working with experimentalists who are the key people to actually inform us on what the function is now. So I think we need a functional genomics initiative now. So as I want to end by thanking, this was just to emphasize as well that we've always believed in fair principles, and especially Ian in supplying them to the community. And also, I should have had a slide, actually. I do have a slide with very many people who took part in the development of CAS, people in the team who use it or add to it. Also, there are collaborators in Cambridge, Tony and Alexi, also Barcott's team and Martin's team, and also Samir's team as well, that we've worked with over the years. But I want to end with this slide. A big thanks to Ian for actually godfather of CAF, I think, keeping it going. But also Nico Boyan, who's done really a lot recently to bring us into the modern age with using deep learning techniques. And for Charlie, who's been a wonderful structural analyst in the team and really gave insights into that pathogen example that I showed you, and Jude for his fantastic chain sort of, but also all of David's team and that wonderful experience we had collaborated. I'll stop there, sorry, probably too much. Thank you. Thank you.
Thank you so much, Christine. It's great to hear about a resource that's been around for 30 years and hopefully will continue for many more.
In some form.
We will now take questions from the audience. And while you're thinking of your questions, I just have a quick question. I know that community input has been very important as you've been developing CAF, and I just wondered if you ever got feedback from the community that changed your plans about what, priorities or features that you were going to add.
So we're lucky because people do write to us, and so we often, we will often adjust things according to what people need to see more of. I think, if I'm honest, the biggest changes to our resource have come through these collaborations we've had with a wider community, like the structural biologists and the experimental biologists in that structural genomics consortium, who said, oh, it's okay having all these families and all these fold groups, but actually we want to know what they're doing. So we need you to tell us, what are the relatives that are doing the same thing functionally. So those sorts of collaborations have helped us a lot. So I see them as a kind of primary community for us. But also we do get lots of requests from the community on changes that we could make to make our tools more easily usable, to make access to the site more easy to use as well. So they've been very important in the development of CAF as well. Thanks for that question.
Okay, we have questions from the audience.
Thanks. Thank you, Christine, and David Parry-Smith from the Sanger Institute. So I'm just interested in, you showed it a couple of times, I think, where you've got the structural conservation residues and then you've got some tunable ones.
Yes.
So is that something that you have to look at in a case-by-case basis, or is that something that drops out of any of the automated techniques?
So actually it drops automatically out of our farm analysis. So we see these highly conserved residues. And especially where we've got multiple functional groups within a superfamily, we start to see what are the highly conserved residues, what are the positions in all of these groups that are conserved, but are they conserved in different ways. And then we have people that we collaborate with, and Vishali Warman, who I've already acknowledged, who then goes in and looks to see, what are these residues likely to be doing? What parts of the ligand could they be interacting with? So I should have pointed out, we've always done manual curation in CAF. So we've always curated very difficult cases where we've not been sure how to assign the domain boundaries, and very difficult cases of remote homologs. So it's a mixture that we've always built we've always built algorithms as well to handle the scale of the data. But then we always have had a person on our team, you know, a sort of data analyst, a scientist who then goes in and checks that the algorithms are working properly. And I think that's absolutely fundamental. You know, these algorithms do not have 100% accuracy. Even AlphaFold, we can see quite a few problems in some contexts. And you have to have curators that go in and understand where the algorithms are going wrong so that you can redesign your methods. So I think that's a really key essential community that's not recognized enough, as you heard this morning.
Yeah, sure. And just an aside comment is that I'm sure a functional genomics program based on these results will be something that the SANG is absolutely interested in.
That's a wonderful thing to hear. So I hope we can persuade someone to fund that.
Yeah,
I mean,
welcome.
I think that
Would
has to be, I
definitely
mean,
be interested
Ido
in
would also be interested. This has to be a key. I mean, we have nearly 250,000 functional families. So obviously we can't target all of those. So what are the key families involved, perhaps in human disease, pathogen disease, you know, which are these key ones that we might want to target with experimental work? And actually there was an interesting discussion yesterday in the fellows workshop. Is there some way we could actually use AI, even ChatGPT or Gemini, to help us work out of these very many functional groups, which are the ones that might be most useful for humanity? So you know, there's fantastic work going on at the EBI in Uniprot and Interpro around using these AI tools to help analyze the different proteins and the family subgroups. How can we use these tools to help guide us into what we should get experimental data from first?
Yes, because we have a flagship program called Multiple Assessment of Variant Effect. I don't know whether you've heard of it, MAVE. We're using saturation genome editing, but to me, having come from a structural and biophysics background, we're looking at the sequence and not hugely thinking about structure.
Well, I think this
guiding
is where,
those experiments.
But we have, you know, we have a huge debt of gratitude to John Jumper and his team, and actually to the, you know, the sort of, it probably is really 30 years of structural bioinformaticians who developed all the tools leading up to that. You know, we really have a debt of gratitude to them, because having that structural data combined, as you say now, with the sequence data, I think can really help people work out what these proteins are doing and which ones are doing different types of things that we can focus on and target first of all.
Hi, thank you very much. You talk about funding. We do currently have some funding from EPSRC to create a multimodal AI network, and we have decided to put a majority of funding into building open multimodal like AI benchmarks. So I think our challenge is to just like the question I asked John yesterday and also Charlotte yesterday, is to find the problem that is ready and also worthy to work on, so that we can bring, I think we have the capability of building good software and bring the AI and machine learning researcher together to work on some problem that if we succeed, it can be very beneficial to the broader community. So
that's wonderful to hear. And I know in the UK, I think Charlotte Dean's been quite instrumental in helping set up this Open Bind project that will solve I think it's 500,000 structures they want to solve together with ligands. So we get information on binding characteristics of different ligands and different kinds of pockets. And that's obviously going to be hugely valuable. Together with the Structural Genomics Consortium also do that. So I think these large-scale initiatives will help bring in the experimental data together with, like you say, the AI that's needed and the kind of domain experts that are needed around structural biology, functional divergence, that kind of thing. So I think there are great opportunities going forward in this area.
But the challenge is that it's such a big, it's more like the big data problem, right? There are so many problems we really don't know, like what problem to work on as
it's the same with the structural genomics, where we were at that point, we were just overwhelmed by the amount of sequence data where we start. but it's the reason why you have to organize it in some kind of coherent way that makes biological sense as well. And then you can narrow down the sort of avenues you want to focus on. And I have to say, it's where you need these assessments like CAFA that are so key for working out whether we're going in the right direction or not.
Yeah, thank you.
Yeah,
so
thanks
much.
for mentioning that.
Thank you for a very comprehensive talk. I have a question about, you mentioned that AlphaFold produces structures that have a high, I guess, component of disordered regions in them, higher than what is in Cath. So my question is twofold. One is, what's your reference structure to that? And the second is, why do you think AlphaFold puts in such a high component of disordered regions?
Well, I think there are very important disordered regions in proteins, and some of them are functionally important as well. I
I'm
mean,
not saying they're not. I'm just saying, what do you think AlphaFold puts in such a large component as opposed to what is expected?
I'm not sure I'm the right person to answer that
Okay.
question. You need someone who's a bit more expert in the algorithm. I mean, I think those are regions that have multiple conformations quite often. And I think you heard last night why AlphaFold sometimes struggles with those regions in the protein. So some of that is because there's uncertainty about the conformation of those regions. And also quite often those regions only become ordered when you bring in the protein partner to which it's binding. And AlphaFold's not using that data. So I think that's perhaps some of the reason why they struggle with those regions. But obviously they're quite good at identifying those regions. I think Silvio Tossato has shown that AlphaFold is probably the best predictor of disordered regions that there is at the moment. So they do very well in identifying them. But I think they can't predict them because it's not clear what their structure is until you have the full environmental context of the protein. I hope that answers your question, but obviously they're key regions.
My question is also, when you say that AlphaFold produces disordered regions as opposed to what's in Cath, so the reference is a crystal structure, or?
I see what you're saying. I mean, perhaps I didn't explain that well enough. It does very well it generates all the highly ordered domain, the globular regions in the protein. And that's the data that we're interested in bringing into Cath. So we can't handle disordered components as well. We don't have the resources. And so we've only focused on those highly ordered regions. So I don't know if that answers. I'm not sure I'm quite understanding.
No, that's great.
Yeah.
No,
Is that okay?
Thank you very much.
So those are the regions that we've really concentrated on with the resources that we have.
Thanks so much. If there are no other questions, let's thank Christine again for a great talk.